With this, I go now to the second part. This is phosphatidylethanol. For phosphatidylethanol, we developed a fully automated method in dry blood spots, which we were able to publish in the Journal of Analytical Toxicology at the beginning of this year. This work was done in collaboration with Wolfgang Weinmann at the Institute of Forensic Medicine, where the developed method is currently in use for routine DBS PEth screening. Phosphatidylethanol itself is a group of molecules which consists of a head group. Attached to this head group, we have a glycerol backbone with two fatty acids at different positions, which can differ in their length and degree of unsaturation. Phosphatidylethanol itself is an abnormal phospholipid, which is usually not present within the human body unless ethanol is consumed or somehow formed within the body. Depicted here, almost in the middle, is PEth 16O18-2. This means that one fatty acyl chain has a length of 16 carbon atoms without any saturation, without any unsaturation, and the second one has a length of 18 carbon atoms with 2 unsaturations within, so 2 double bonds. Considering the formation of phosphatidylethanol within the human body, we have to have a look at this figure here. Phosphatidylethanol is formed within the human body when phosphatidylcholine loses its choline head, which is a natural process which is occurring due to the presence of phospholipase D. This process is normally forming phosphatidic acid, which serves as a second messenger within the human body. However, if ethanol is present, ethanol acts as a very good nucleophile within the enzyme and causes the formation of phosphatidylethanol. As already mentioned, PEth itself is a group of phospholipids and not just a single molecule. PEth 16O18-1 and PEth 16O18-2 are the two most abundant forms of phosphatidylethanol being present in human blood and are often measured together. PEth itself has a half-life of 3 to 12 days. It is not stable in liquid blood due to the fact that there are enzymes degrading it unless that blood is stored at -80 degrees Celsius. Therefore, it was found that DBS is the method of choice for the analysis of phosphatidylethanol, as when dried onto the DBS card, PEth becomes stable for a much longer duration. Considering the time window and the detectability of direct alcohol markers such as PEth, we have to have a look at the following figure here. It is like that you can split up the direct alcohol markers considering the temporal window of biomarker detectability on the x-axis and the lowest detectable level of alcohol consumption on the y-axis. Here we can see that blood alcohol, breath alcohol, and urinary alcohol are already detectable when very low amounts of alcohol are consumed, however only for a very limited duration of time. Urinary EDG and urinary ETS are detectable for a little longer, however still not long enough if you want to investigate if somebody is abstinent or has an alcohol problem. And this is where phosphatidylethanol comes into play, as it is detectable for several weeks already at low to moderate levels of alcohol being consumed. For longer time windows, hair analysis such as fatty acid ethyl esters or EDG can be used. However, hair analysis has the problem that the hair has to grow out of the head first, so the last 2 weeks right before the sampling are usually not covered by hair analysis. Phosphatidylethanol within the human blood is believed to be, or is most likely part of the red blood cell fraction especially the cellular membrane, as phosphatidylcholine is really abundant there. This is a figure from the USDTL lab, which shows that quite nicely. There have been a pharmacokinetic model being developed, which show a little bit how phosphatidylethanol over time forms within the human body. This is depicted here on this figure. This figure shows the PEth concentration increasing during 21 days, followed afterwards by alcohol abstinence. On top, we can see what happens if every other day 3 drinks are consumed over a 2-hour period. Then in the middle, every 3rd day, and in the end, every 5th day. On the left, it's depicted for male subjects. On the right, it's depicted for female subjects. What we can see that over time, when consuming regular amounts of ethanol, the phosphatidylethanol concentration reaches a plateau level where the formation and the elimination hold itself in an equilibrium. Once the consumption of ethanol is stopped, we have a decay with a certain half-life of phosphatidylethanol within the human body. The application of phosphatidylethanol in real life are various. One example is, for example, during driving aptitude assessments. This is performed also within Switzerland. If somebody gets caught with a too high amount of ethanol within their blood, they are assessed if they are fit to drive, to participate in traffic. A statistical evaluation based on rock curve analysis came to the following threshold, that below 20 nanograms per milliliter of phosphatidylethanol, we can speak about alcohol abstinence or minimal consumption. That between 20 to 150 nanograms per milliliter, we have moderate alcohol consumption, and above that, we have generally excessive alcohol consumption being present. Another application where phosphatidylethanol proved to be really a key playing compound is alcohol withdrawal and dishabituation therapy. Here depicted for subject number 6 from a real case study which was performed in Switzerland. We can nicely see for venous DBS, PEth 16018-1 and also 16018-2, that once subject is abstinent, the phosphatidylethanol concentrations decrease over time. It can also be used to detect relapse during the alcohol withdrawal and dishabituation therapy. Here depicted for a real life sample or situation of subject number 4. This subject was abstinent for about 40 days, and afterwards the Christmas holidays took place, where we can see high rises in PEth concentration in venous and capillary blood. We can see here that once the subject started drinking, the phosphatidylethanol concentrations remained high during each of the sampling time points. In parallel, EDG and ETS in urine were sampled. However, we can see here, due to the shorter half-life of this marker, which is up to about 24 hours only, that at least at 3 time points, the concentrations were very close to the LCMS cutoff concentrations for ETG and ETS of 0.1 micrograms per milliliter. However, if these 3 samples would have determined by, for example, a ETG immunoassay, they would easily have gone missed due to the fact that one would not have detected the very low amount of ETG, which would somehow indicate that some alcohol consumption has happened.